Good afternoon to you all. Now we are going to start the second technical session of the day on shrimp health. I invite Professor K.M. Shankar and Dr. Devika Pillai, Madam, who are the chairperson of the session, to come on the dais. I request Dr. Devika Pillai, Madam. I also request Dr. Kalaya Situnya Luksana Dangtip, who is a keynote presenter, and Dr. Arun Kumar Dhar, the lead presenter, to come on the dais. I also request Dr. T. Satish Kumar, Senior Scientist of ICR SIBA, and Dr. Jena, Senior Scientist of CIFE, to coordinate the session. For technical session on shrimp health, we have the chairperson, Professor K.M. Shankar, who was the former Dean of the College of Fishery at Karnataka Veterinary Animal and Fishery Science University, Mangalore. His translational research has led to successful transfer of technology like Rapidot kit, which is a monoclonal antibody-based kit for white spot syndrome virus in shrimp. Similarly, he has also developed artificial substrate-based technology to enhance the carp and tilapia production through biofilm formation. And this he has been able to successfully disseminate it to the farmers with the help of UNESCO and NAIP program. Professor Shankar has received numerous awards and recognition, including the award Bharat Ratna Dr. C. Subramaniam Award as a outstanding teachers in 2008, and Biotechnology Product and Process Award from DBT in 2012. He has also won several dozens of other recognition and professionals from the different professional societies and university. He has been able to conduct several research projects funded by the different research organizations like DFID UK, ACIR Australia, European Union, and UK-based agencies. Our second chairperson of the session is Dr. Devika Pillai. She is currently serving as the Assistant Director General of Inland Fisheries in the Indian Council of Agriculture Research. Formerly, she has been the Director of Research at Kerala University of Kufos, and she has made a significant contribution in the fields of fish pathology, disease diagnostics, AMR, and aquaculture systems. Dr. Pillai has played a pivotal role in formulating national strategy for aquatic animal health management. Her work has advanced the understanding of host pathogen interaction, disease surveillance, and therapeutic interventions in cultured fish species. Notably, she has been instrumental in promoting biofloc-based aquaculture technology in India, enhancing both productivity and biosecurity in inland fisheries. Now I request the chair to conduct the session. Thank you.
Welcome to the session 2, Shrimp Health. Ladies and gentlemen, we have first a keynote address, followed by a lead presentation, followed by 9 presentations, a long session. First, we'll go with the keynote address by Dr. Kalaya. I'm using the first name. Dr. Kalaya is a leading expert in shrimp health and aquaculture, serving as principal researcher and head of the aquatic animal health team at Biotech NSTDA, Thailand. Recognized globally, including the Newton Prize Chairman's Award and Stanford Top 2% Scientists in Fisheries. Dr. Kalaya leads the Shrimp Immunology Platform at Maidal University and contributes to international initiatives like the International Network for Shrimp Health, NSH. Her research focuses on shrimp pathogen interaction, disease prevention, and genetic improvement of black tiger shrimp for growth and resistance. Her latest work explores viral accommodation mechanism to develop shrimp vaccines and enhance viral tolerance in breeding programs. The topic is application of scientific knowledge to control EHP infection in shrimp.
Hi, hello. So I'm the first one after lunch. Hope everybody is still okay to listen to my talk. Okay, so first of all, I thank the DAA 12 organizer for inviting me here. And I hope I can share you with our scientific result with the EHP recently during this 2024 and 2025. Let me start by introduce a little bit where I come from. This is my agency. We are under the Ministry of Higher Education, Science, Research and Innovation. And my agency is National Science and Technology Development Agency, NSTDA, or you can call us NASDA. We are in the Thailand Sci Park in Pathum Thani province in Thailand, not Bangkok, Pathum Thani. And under NASDA, we have 4 national centers that we are working together. The first one we call our Cell Biotech, which I come from, National Center for Genetic Engineering and Biotechnology. Then we have the other national centers, including National Metal and Material Technology Center, National Electronic and Computer Technology Center, National Nanotechnology Center, and National Energy Technology Center. So we have totally of about 2,000 researchers and 600 of a PhD level, and working together in the, the common interest topic. For aquaculture, our focus will be measured in these 4 disciplines: precision farming, animal health, environment, and feed. So biotech, which is my agency, we are going to work a lot with this, this group, the animal health, and also with this live feed and microbiome and AMR. And we also working with the other national center to work on this precision farming, such as IAS, Aqua IoT. Our aim in 2028 is low carbon aquaculture system for the sustainability. So as a director of the aquaculture group, I will come over the collaboration to lead us, help us, support us to go to this 2028 mission. And NASDA also value the collaboration from all the agencies, such as the International Research Agency, university, the research from abroad and national, industry, policymaker, and governmental agency. Okay, if you want to know more about us, please, this is our website, so you can look at what kind of research we have in NASDA. Okay, I will start by telling you about, this is the last review that we have been written about EHP. One is in the Journal of Invertebrate Pathology 2021, and another in, as Dr. Vitalia, as an editor of this shrimp book too, in 2022. So what happened in terms of research from our group after that? Okay, this is what we know so far about EHP, or the research from our group and the collaborative team. We start to know that it has a horizontal transmission by using this model, the cohabitation model. We know the problem is the spore. We can purify the spore, and then we can induce them to to perform this polar tilt shooting. We know that why they need the energy, or why they make the host growth retardation, because they cannot produce the energy themselves. So because they lack a lot of glycolytic gene from the Mycosporidium genome that we have. We know the mechanical carrier of this EHP. We use the result that we got to for the farm to use, for example, the high pH during the pond preparation, just to let the spore that left in the previous crop shoot, and then they die. We recommend some disinfectant that can be used to disinfect this EHP, the temperature-dependent spore shooting activities, blah, blah. The PCR method that we recommend you to use them, and the method that we recommend is spore wall protein primers. We understand the pathology of the EHP with a lot of developmental stages in one cell, and also what happened in the when spore get into the intestine or in the digestive tract of the shrimp. Last but not least, we suggest that you should treat the water. You cannot release the water directly to the environmental reservoir if you found that your ponds or your shrimp get infected with the EHP by very simple resting them in the pond for at least 10 days. So this all of the work that we have done before 2024. And the point is, until now, we still heard that many farmers still get the problem with the EHP. That means our scientific study is not finished. So we go on. Okay, this is from the study, this is what we recommend for the farmers, from hatchery to the waste treatment ponds. I will not go through this all of this, but we can talk if you are interested in which method from the scientific knowledge that we have can be applied in each step. What we would like to point out with this EHP is that when you said positive PCR, it doesn't mean disease. This is, if you have positive PCR for white spot, you got white spot disease. But if you got positive PCR for EHP, the shrimp will be or will not be diseased. It depends on how strong they are at that point. And the cutoff of this EHP is about 10 to the 3. From our result, 10 to the 3 copy per nanogram DNA. At this point, if higher than this, disease. Lower than this, we have chance to work with them, so they cannot get disease. Okay, so all of these application or strategy that I'm talking in the previous slide, we have worked with them in our demonstration farm in Thailand. First, you have to have the first, what you call must, is that you have to have not very high stocking density. After that, EHP has to be checked in the PL and also during the culture. So this red box here is before we apply all the strategy to this farm. You see that they did not do any treatment, so the EHP going up and up until you get 100% in the pond. And this is how the weight of the shrimp like. But when we apply this, they can maintain the level of the EHP during the cultivation, and this is what we saw in the farm. The weight can be going up. Okay, so today this is what I'm going to talk about. During 24 and 25, this is what we do. We think that it might be the PCR method, especially during the DNA extraction, that make the detection is not effective enough. So we collaborate with Professor Huang Ji at Yellow Sea Fishery Research Institute, and we found that this method to put chitinase and proteinase K in the lysate buffer for PCR for infected tissue extraction might be effective. I'll tell you about that later on. And we would like to know more if EHP cannot make shrimp slow growth by not having this glycolytic enzyme, and what they do after they infect into the shrimp cell. If you remember, I was very interested in this story for a long time. How can EHP going into spore of the EHP with the size of 1 to 2 micron, going into the stomach of the shrimp, how can they pass the gastric sieve, which have the size of only 0.75 micron, to the hepatopancreas? So today I think I know, so I tell you. Midgut might be another way of the EHP spore to go instead of going from the stomach to the hepatopancreas. And then we found one protein is very, very interesting. We call, this is working with Dr. Andy here, in way with the question of if there is any vertical transmission of the EHP. And from our study, we still think that no vertical transmission. One protein of the EHP that we found, we call it polar-2 protein 2, which is very important protein during EHP spore shooting. When we study more in the detail, we found that we can use this polar-2 protein. We can inhibit this polar-2 protein, and then the EHP cannot replicate. The way we inhibit it, we use what's so-called anti-PTP2 antibody. I'll tell you in detail later on. And to make it cheap, we produce them in the egg yolk, hen egg yolk, or in the form of immunoglobulin Y, IgY. Lastly, I'll touch a little about what we have done with this EHP white feces syndrome. In our group, we divided white feces syndrome in several categories. One is caused by vibriosis. I think many people know about it. But in our lab, the work that we have done, we done with the white feces which have the EHP as a component factors. So we call it EHP white feces. Okay, start from extraction of this spore of the EHP. If anybody working with the PCR, maybe this method can help you to improve the DNA extraction. So I just teach you a white feces, you add chitinase first, and then after that, you add proteinase K, and then do the normal PCR extraction. That way, this is the result. If you have not have anything, this is the copy per volume per microliter. You get, if you add proteinase K and chitinase CP here, you will see that the number of the copy is increased. You will see more when you look at the copy per spore. With this chitinase and proteinase, you get almost many times higher of the copy released from the spore. So I recommend this method to be used for the PCR extraction. And this is the work we done with CFAST team and University of Exeter. And the result showing us that the EHP doesn't have the glycolytic gene. So it means that they cannot generate ATP itself. By that, what did they do? This is our new result. We found that EHP has 4 energy transporter, or we call it nucleotide transporter, to help them to transport the energy from the host into EHP for replication. And if we inhibit this EHP by double-stranded RNA, you see that the replication rate is reduced significantly. So now we start to understand more how can we, protect or inhibit this EHP replication. Besides, when we stain the hepatopancreas of the EHP-infected shrimp with the oil red ore to stain the lipid, we found that this is control. You see that the lipid is everywhere, especially in the B cell of the hepatopancreas. But when EHP infected, this is what happened. The red droplet is significantly reduced. That's why they cannot grow very well. Okay, this is what I'm talking about. We published this paper in 2019, and to our surprise, the gastric sieve, which is the area between the stomach and the hepatopancreas, have the hole of only 0.75 micron. So we would like to know EHP with 1 to 2 micron of the spore size, how can they infect the hepatopancreas? Okay, this is the normal way. This is the normal pathway of the food going into the digestive tract, the mouth, and then go to esophagus, goes to stomach. And then during the stomach, like I said, we have the gastric sieve. And this gastric sieve will allow this fine material, less than 0.75 micron, going to the hepatopancreas. How's about the coarse particle that bigger than 0.75, they go to the midgut. And this is the result that we found, that the EHP is also going to the midgut. This is what it looked like. The gastric sieve is here. So from the stomach going to the hepatopancreas, and the coarse material is going directly to the midgut. And this is what we found in the midgut, a lot of PCR positive EHP. Okay, so in this sense, I would suggest that when you have the hepatopancreas, you want to detect the EHP in the hepatopancreas, use the whole hepatopancreas for DNA extraction. You get the midgut along with the hepatopancreas. Okay, the vertical transmission, I think I have read several papers, but I'm not sure the way to collect the sample, especially from the ovary. You see that when we design the experiment, we have this dot area here. We use it for in situ hybridization, but for the PCR, we'll use this area because we don't want we don't want the sample to be contaminated with the HP. So we have to divide them into pieces. When we collect the sample, when we check, we found that we see the EHP only positive in the hepatopancreas, not in the ovary and not in the testes. Both in several places of male reproductive organ like testes, vas deferens, and terminal ampoule, we don't find any positive signal so far. So if you want to study this, maybe you have to be careful how to collect the sample. The ovary is very sticky. If you collect them close to the hepatopancreas, you might get the contamination of the DNA from the hepatopancreas. Okay, this is another project. Like I said, the protein called PTP2 is interested us because when we try to understand the function of it, it turned out to be that this is the way we study the PTP2 in the B is HP, and C is intestine, and A is stomach. We don't find PTP2 in the stomach at all. It means that the polar shooting process is not occur in the stomach, but in the hepatopancreas, more than 95% in the hepatopancreas, and some in the intestine. So when we try to knock them down, not let them express, it seems like if you don't have this PTP2, then the EHP cannot replicate. By that, we produce them in the egg yolk, the hen egg yolk. We call it AQ35 because the size of this antibody, the size of PTP2 is about 35 kilodalton. Now we already success in produce it in a large amount. And when we use it in our demonstration farm, you will see that this is the first test. The number of EHP reduced to the level that cannot cause trim growth retardation. Or this is the second test, the result still confirmed. So at this stage, we send them to the farmers to test them by the protocol they are using in their own farm. Okay, I will talk a little this YFC syndrome in my team, we always found that the samples sent to our lab reported YFC syndrome always have the EHP involved. And it seemed to me like if your shrimp got EHP infection, it will go two ways. The first way is the EHP replicate and then cause the shrimp growth retardation, or during the EHP develop, then there is a bacterial infection, maybe because of the excess organic matter in the farm, and then the bacteria grow, then the bacteria infect in the shrimp that already infected with the EHP, then you will find this YFC syndrome. And when we look closely, we found one Vibrio, we named it 6HP. 5HP is Vibrio appen in our lab, which is very, very potent. And 19HP is non-virulent Vibrio. You see that this is on the y-axis, this is the number of shrimp with YFC syndrome. And this is the day post 6HP challenge. It's only 6HP here that caused the high number of the YFC shrimp. Okay. Whereas 5HP can do the same, but since they are EMS in its appen, so they die before they can go further. So we are very interested in this 6HP, and at the same time, Dr. Luis Alangulen, they published this paper, and it seemed like the way they are working or do the experiment is more or less like we do. Okay, the YFC shrimp, the induced YFC shrimp in the lab show the pathology similar to what happened in the farm. For example, they lose exoskeleton, they reduce feeding, retarded growth, high size variation, and high FCR, and sometimes mortality. So when they start to get the YFC, they don't eat. So you see a lot of feed left in the tank like that. No? And if we don't do anything about it, you keep them like that, the mortality will come. So it's exactly like we see in the farm. This is a pathology of the hepatopancreas. If you, in the first day of the YFC, you start to see thin tubule, thin epithelium tubule, and then you let them go further, they start to have this sloughing, and then now all systemic infection, you see this, a lot of encapsulation, and shrimp will die on this day. So when you start to see YFC, which is in this case day 1, they still have the time for you to do something about it before they develop into YFC day 6, which you cannot do anything about it, they will die. Okay, so this is what we conclude about the YFC. It associates with the VP appen. We call it 6HP or chronic VP appen. This is a little bit contrast. Appen is acute hepatopancreatic necrosis disease, but this one is chronic. You have to, give them, and then they will develop the YFC later. So infection of EHP will result in more susceptible to this 6HP. And YFC is the result of hepatopancreatic cell sloughing, disintegrated from the tubule, and then released in the epithelium tubule. Then you see it as a YFC going out in the intestine. The faster detection of YFC, you have chance to recover the YFC. They can come back and still be able to feed again. But if you let them go, they will have the high damage degree of the HP, and then that cannot recover, not reversible. So now we have this EHP YFC laboratory model, and then we try to use them to study if we have the inhibitor or anything to stop this YFC syndrome. So last but not least, I still think that working with the disease in shrimp or aquatic animal, we need a collaboration. So the network is very important for me. If there is any information that we can share, it will help us to solve the problem very fast. So this is all the collaborator and granting agency. I have a special thanks to team CFAS and University of Exeter, that the first time we are working with EHP, we got a very high support for them to solve the EHP problem in Thailand. And this is the group of the private sector that we are collaborate with to deal with, to try to inhibit this EHP and YFC syndrome. This is the team, and also my team at Centec Trim. This is at Biotech NASDA. We also have this, what you call it, kind of center, aquaculture center. We call it functional feed innovation and testing. And we have many researchers from many disciplines to help you to design the experiment if you like to, test some product of the aquaculture commercial products. Okay, thank you very much. So, no question. Oh, 5 minutes before, so I can have several questions.
Yeah, thank you for this very interesting talk. And it's interesting to know that there is a AHPND VP associated with this white feces. But it's chronic, you said. It's not causing an acute AHPND. Now, is it due to any difference in the expression of Pyrrhae and Pyrrhae? How is the expression compared to the normal strains which cause AHPND? So what's the difference between this chronic and the acute AHPND strains?
I would say the difference between 6HP, the chronic VP and the VP appen is that the VP appen will produce the toxin, both per A, per B, very quick. Let's say on the day, start from day 1, 24 hours, they start to produce the toxin. But this 6HP, they will produce late, very late. Let's say 5 days after the infection, something like that. So for me, it's kind of perfect working with the EHP. When the hepatopancreas cell got weak, then the toxin will come. So that's why the cells sloughing and then cause this YFC syndrome. So the recovery will come when you kill this VP appen, VP6HP. So if the way we do is very easy, we just change the water immediately, and then lower the stocking density, and then they will sometime they will start get fed, and then they will come back.
And this delayed expression of Pyrrhae, Pyrrhae, is it only in vivo or in vitro? Also, you find the same delayed expression.
Sorry?
You said it's a delayed expression of Pyrrhae, Pyrrhae, because it comes late, you said. So is it only in vivo in the animal tissue or in vitro also? If you try to express it in vitro, is there a delayed expression, and is there anything known why there is a delayed expression in this?
What we have done, we only did it in the tube. We grow the bacteria, and then when it comes to the same number of cell per ml, then we check the per AB concentration. But I mean, this is corresponding to what we see in the histopathology of the shrimp too, that you see the appen pathology, the cells sloughing in the YFC pretty rate compared to this happen, the violence happen.
Thank you very much, Kalaya, and to your team for this very in-depth research on EHP. I think the shrimp is in trouble because it's aside from EHP, you have the append, which is one of the most significant non-viral disease. You have mentioned earlier some measures to control or manage, but in terms of prevention, what sort of practical measures could you advise? I think there are many farmers here. Which do you tackle first, the EHP or the append, which is a bacteria? So I think that the private sector will immediately use some antibiotics. So some practical measures, yeah, based on your understanding, very deep understanding of EHP and AHPN. Is it clear?
Yeah, actually, we are aware of this. There is several vibrosis now. You might also heard about TPD, GPD, pale disease, blah, blah, blah. So this is also our concern in the team. So this AMR issue was raised, and then we got a grant from IDRC from Canada to work on this, that are there other alternatives to this antibiotic for this vibrosis in shrimp. And now we come with several nature-based solutions. Ed might like this word, no? Nature-based solution. For example, we have phage. We are working on phage cocktail, and we are working on the immunostimulant, which have already tested to be able to inhibit the Vibrio infection in the hatchery, things like that, to help, as an alternative for the farmers to use, and also make sure that they understand the risk of the AMR.
Mr. Chair, can I have a question? Kalaya, thank you. There's a lot of basic work that has gone into what you're presenting, which is very essential for the practical applications to be thought of later. My question is, see, in all kinds of syndromic problems that appear in the shrimp industry, there is a succession that we see. Now, in your studies, have you tried to look at the succession of organisms? Which comes first, which comes later? Does one predispose for the second or for the third to come in, That is one thing. Because the gene interactions would be very important in deciding how one pathogen predisposes and the propensity for the second one to come up, and then third one. Like in many syndromic conditions, it's like how one yields into another, no? So we have seen the association of Vibrios with many, many syndromes. So white spot syndrome was one of them, and invariably Vibrios were associated. In fact, at the Central Institute of Brackish Water Aquaculture, they have been studying in an earlier conference that I've attended. I've heard the presentation about the succession. And in your studies, could you please enlighten if you have looked at what comes first and how does it help the second and the third to be there, okay? Thank you.
Okay. In my opinion, there is only one co-infection that it really is, which is white feces syndrome, in my opinion. And this is because two pathogens, they cause different outcome. For example, the EHP, because they cause slow growth, they cause, it takes time for them to cause the weakness into the shrimp. And for me, it's making sense that it will, finally the opportunistic Vibrio will come and cause something to them. And in this case, white feces syndrome. But in the other cases with several viral infection, multiple viral infection, in my opinion, I might see it, but I would not say it multiple infection because the first one is already non-violent. For example, they live happily together. I would suggest IHHNV. You go now, look at the sample with IHHNV positive. You see IHHNV inclusion, but no problem of slow growth, no problem of, I cannot remember the name of this. Okay, no problem anymore, but positive, and you see the virus there. And then that shrimp got another infection, maybe by white spot, which again, the different outcome, IHHNV growth retardation, and then white spot, death, mortality. So if you are going to see the multiple infection, you will see only with this kind of pathogen, with different outcome. But you will never find Thorla syndrome virus with white spot for me at the same time because the outcome will be the same. I don't know if I answered the question, but so far I don't see myself. Okay.
Excellent. Yes, sir. Thank you for adding one more confusion. Already we had a lot of confusion, ESP, white fecal, so many. Now you added chronic AHPND. So one more confusion you added into the whole confusion already existing. So have you isolated any time this chronic AHPND in the absence of WFS or white fecal or EHPND?
I'm sorry, can you ask again?
Have you isolated this chronic AHPND independent of all other complications?
Do I have the isolate of 6-HP in my hand?
Is it available?
Yes. No.
No, is it always come with the white fecal or it is independently, it is isolated? Chronic AHPND you told, no?
You mean I isolate this chronic AHPND from the shrimp?
Healthy shrimp? Any other shrimp other than white fecal where there is no other complication?
No, not with other healthy shrimp, no. But it might be already in the pond, you never know, with the small numbers. And whenever the shrimp weak, then they can infect shrimp easily. I would think that it's already in the pond.
1 minute.
Okay.
Hello, Dr. Kalia. That was a beautiful presentation. My question is, what impact the salinity and organic loads in the pond have on the severity of EHP and white feces? That's my first question. And the second one is Penase monodon refractive to these two problems? Because we find these problems much less in black tiger farming, which is being currently undertaken. These are my two questions.
Okay. This is a good question because I have an answer. I can answer the second question first. We have one master degree student working on that, and Penase monodon has less susceptibility to EHP, very less. And Macrobrachium, no problem at all.
Yeah.
Okay, that is second question. And the first question, for me, what I saw is that if you have, like I said, if you have EHP and then the bacteria come, your pond will have the problem of white feces and shrimp start to die. But if they don't have the bacteria in the pond, the shrimp that infect with the EHP, they will not have problem with the survival rate, but they will keep slow growing. So it will be two-way like that. You will not have both of them in the same pond.
Okay, how about the salinity, impact of the salinity? Because the lower salinity and higher salinity, does it have a difference?
We haven't done that.
Okay. And the organic loads directly, in case of the pond water quality being good with less organic load, how does it reflect on the severity of EHP? Obviously, your append is going to be low, that is non-PRAPB 6HP, what you said. It's going to be low, so will it have a...
Less organic load and less white feces syndrome.
Right. Okay, thank you.
Okay.
Thank you, Dr. Kalaya. Nice presentation.
Thank you.
Before the...
Yeah, good afternoon. The next we'll have the lead presentation by Dr. Arun Kedhar on reverse genetics approaches in developing oral vaccines and therapeutics in fish and shrimp. Let me introduce the speaker. Arun Kedhar is a professor and director of aquaculture pathology laboratory in the School of Animal and Comparative Biomedical Sciences in the University of Arizona, USA. Dr. Dhar is a World Organization for Animal Health expert for crustacean diseases and the Aquaculture Pathology Laboratory in the University of Arizona is a WA reference laboratory and an USDA APHIS approved laboratory for diagnosis of crustacean diseases. Dr. Dhar leads efforts to provide disease diagnostic services to the shrimp industry worldwide. His research focuses primarily on pathogen discovery, developing diagnostic tools, and oral therapeutics and vaccines against viral diseases in shrimp and fish. He serves as an associate editor of the Journal of Invertebrate Pathology and reviews in fisheries science and aquaculture. He also teaches at the Cornell University Aquavet program as an adjunct professor. Sir, the floor is yours. Thank you.
Good afternoon, and thank you so much for your kind introduction. First and foremost, I would like to thank Dr. Kuldeep Lal and the organizing committee for inviting me. I'm so happy to be here, really. I always feel at home coming here. What I'm going to do is to give you an overview of some work in our lab. These are ongoing work, highlighting how we are trying to develop a viral vector that could be used for a wide variety of molecular works. So with that, let's look at a quick outline. So first, I'll give you a very quick overview of what are the challenges that we face while trying to develop reverse genetic approaches. And the second, I'll give how we developed an infectious clone of an RNA virus of shrimp. And then how we went on to modifying that particular virus to use it as a vector for delivery of therapeutic RNA via oral route. And next, then the last aspect is this approach broad enough that we could use it for fish and develop oral vaccine? And some concluding remarks. So as you all know, the flow of genetic information in a cell goes from DNA to RNA, and that encodes biologically functional protein, what is we call the forward genetics. But today, we have tools and technologies to use synthetic biology approach to change to do some mutations at the RNA level, and then go back to look for the phenotypes, what we call as a reverse genetics approach. Now, we all know one of the major challenges working on shrimp diseases is the lack of a crustacean cell line. However, over the past few years, there has been a lab here, and Dr. Bright Singh is the PI in Kochi in India, where they have developed an hybrid cell line of Penaeus monodon and insect cell line SF9. And this is the first report for production of an invertebrate hybrid cell line. Now, while this cell line is good enough to do some basic virology work, but it has its own limitation. And you can see that, as I copied one sentence from their abstract of the paper published in Virology, basically, when they infected these hybrid cell lines with white spot virus, it doesn't form mature virions. And why and what not, we can discuss all of those. But nevertheless, so we have something to start with, but this is not the best cell line that we have at our disposal. So that's really is a handicap for all of us working in the field of molecular biology, molecular virology, and to understand molecular pathogenesis of anything, whether it's a EHP or any virus, fungi, you name it. To overcome these conundrum, a few years ago, working with colleagues in Mahidol and Tokyo University, Japan, we produced infectious an RNA virus called Macrobacterium rosenbergi nodavirus in SF9 cells. And the way it was done is, we clone, now mRNV is a bisegmented RNA virus. That means it has two RNAs, RNA1 and RNA2. The RNA1 encodes the RNA replicase or RNA-dependent RNA polymerase, and RNA2 encodes the capsid protein. So both of those genes were cloned and in vitro transcribed, and then you went on to transfecting SF9 cells. When you do the transfection of SF9 cells, those cells not only expressed, retained that RNA, but also produce mRNV. Then we can purify that mRNV from the SF9 cells, and then when you use that to challenge healthy shrimp, you can reproduce the disease. And mRNV is known to cause muscle necrosis, and you can see here on the bottom left panel that it does cause. So you have a synthetic virus that was produced via transfection of SF9 cells using both RNA1 and RNA2. Now, while these things looks very interesting, but really this is not a robust method by any means. The paper was published quite a few years ago, but as I mentioned, still the challenges remains. Because if you want to do any genetic modifications at an RNA level, it's not easy. It's really, really difficult. To overcome this, I came up with an idea using baculovirus expression system. There are existing commercially available dual baculovirus expression system, and the system basically contains dual promoters. You can put RNA1 under the control of one promoter, and in this case the polyhedrin promoter RNA1, and under the second promoter P10 you can put the second RNA. So you have a single construct that carries both genes, RNA1 and RNA2. And you can take these plasmid DNA, make backmids, infect SF9 cells, and you can produce baculovirus. This is nothing rocket science today. It's all done. People do it routinely. What is interesting, when you take that baculovirus and infect SF9 cells, the baculovirus replicates, but it also produces mRNV. So because the backmid carries the mRNV genome, the full-length genome, so that means the virus RNA is expressed, the mRNV RNA, and also packaged to make mature virion. Then you can take that virus and infect shrimp, and we could reproduce the disease. And we also expressed another small RNA virus that remains associated with mRNV, the extra small virus, and you can do all kinds of combinations and permutations to make different types of constructs. So this has opened up a whole new avenue for genetic manipulation of RNA virus of shrimp. The paper was published in the journal Virology a few years ago. With that being done, then I realized that you can now, in the same construct, you can remove the RNA-dependent RNA polymerase, and my goal was to put, just for proof of concept, can we put a marker gene? So we replace the RDRP with an GFP, the green fluorescent protein gene, and then do rest of it. So you end up, so what you do, what is the, I can't see. It would have been good to see the, here. Is this? Oh, it doesn't, okay. In any case. So as you can see, we replaced the RDRP with GFP, and then did the same drill. And when you infect the SF9 cells, which one is this? Oh, this one? Okay. I can't see it properly. Anyhow, oh yes, it's. So you can make both virus in any case. So again, you will have the mRNV and the baculovirus. Then I was interested to see, can we purify both of them, do electron microscopy, and mix that cell culture biomass with the commercial pellet, feed it to shrimp, and see what happens if can we express GFP in shrimp. And when we did that, first, we could see very nice expression of GFP in SF9 cells. And the transfection efficiency was very high. That is nothing surprising. It's not a big deal, frankly speaking. However, there's one important message. And the message is the payload has been expressed and folded properly to make a biologically functional protein using a shrimp viral vector. That is very important and very encouraging. Then, if that is true, if you take the SF9 cells and you do transmission electron microscopy, theoretically, you should be able to see both the baculovirus and the mRNV. And sure enough, we could see both the baculovirus and mRNV, which generally forms within a vesicle. And you can see the size, which is about 37, 38 nanometer size. So that means we have a recombinant baculovirus carrying GFP in the backbone of mRNV. Perfect, thank you. And you can see baculoviruses, which are long, elongated type of particles, and you can have the mRNV. These are modified. Now, when we purified, we could see the mRNV and also the baculovirus. So now you know that the SF9 cell is expressing both. Then we went on to feeding shrimp just a commercial diet, or we mixed the SF9 biomass, or we just injected the recombinant mRNV as a proof of concept and see what happens. So when we did that, so we fed for 5 days, or we did only one injection, and after 5 days, we sacrificed the animals and collected hepatopancreas or pleopod, and you can see the titer of the this was measured using the second RNA, RNA2 of mRNV, keeps on declining. That was extraordinarily encouraging for us. That means once you withdraw all feeding, then it slowly goes away. That is exactly what you want when you are trying to vaccinate or deliver a therapeutic molecule. You don't want that molecule to be circulating into the system. You can also, another conclusion is it is a spreading, so it could be absorbed and systemically spread to different organs. However, it does not cause any muscle necrosis, otherwise, which mRNV would otherwise cause, because there is no replication of mRNV. It's a replication deficient virus that can circulate but doesn't cause any clinical pathological manifestations. So we have a wonderful vector. If that is the case, so is that recombinant virus being absorbed by the hemocytes? So we did some electron microscopy, and you can see this is loaded with mRNV. So that means mRNV, when you administer through the diet, is being absorbed, and you can detect it. And if all of these are correct, the hemocytes should be glowing, provided the payload is expressed. And that is exactly what you can see here. Injection was our proof of concept, and oral is our main target. So you see, we have a delivery system, a viral vector of shrimp that can deliver a biologically functional gene and that can express it. This was published in the journal Proceedings of National Academy of Sciences recently. Now, with that being said, we are interested to see, can we do the same thing for shrimp RNA, for fish RNA virus, and VNN, the viral nervous necrosis, is a very similar, it's a different cluster or clades of the nodavirus. And we basically did the same drill just to produce infectious VNN in insect cell. Again, we cloned it. We also made another construct just to replacing the RDRP of VNN with GFP. And we did the whole drill just to show that, yes, indeed, we can produce. And then when we took the cell supernatant from the SF9 cells and infected the OMB cells, and now it's the RGNNV, basically the VNN genotype. And you can see this, the cells starts detaching and cytopathic effect, and you can see the, even infecting the OMB cells with 1-to-1 MOI, it kept on increasing. That means we have produced an infectious fish virus in insect cells, and that can infect cells, of course. Just to make sure that the infectious virus, VNN, is retaining the entire genome, because that's very important. So we made a series of overlapping PCR, nothing but to show that the entire genome, the RNA1 and RNA2, are being retained in the packaged virion. And the data absolutely, confirms that finding. And then when we went on to injecting striped bass, it's a hybrid striped bass. It's not very overly susceptible to VNN, but that's all we had at our disposal recently. And we couldn't detect the virus in gills, liver, kidney, or spleen. However, in brain, we could detect it, and also in the intestine. It's not heavily susceptible. What was interesting, you can see on the left is the healthy fish, on the right is the infected fish, injected SF9-derived fish virus, and you can see basically in the spinal cord, brain, and eyes, which are the target organ of VNN, the histopathology clearly shows that we have produced infectious VNN in insect cell. Now, you might wonder, who really cares, green or modified VNN or mRNV? So what can we do with this? It opens up a whole new horizon. Now, you can replace GFP with a hairpin RNA. You can express any number of payloads as long as the virus can carry. So in other words, if the RNA is big enough and at the same time right size to be packaged, you can package in for fish, for shrimp. You can also modify this VNN. You can genetically truncate it to make it a replication deficient virus. You can make bivalent vaccine, replacing the GFP with whatever you want. Those are the works are currently ongoing in our lab. This has opened, in my judgment, a whole new horizon for developing oral vaccine, all the way to for fish, shrimp, and also for genome engineering. Bear in mind, this is a systemic virus, VNN, or mRNV, and in mRNV, there are papers that it is transmitted vertically. And I'll stop here because you can speculate where it is heading now. All of these bring me to these concluding remarks. Losses due to diseases are inevitable, particularly as the industries, shrimp, fish, is expanding globally, and movement of animals from one corner to another, and so many other factors. We all know it. Prevention alone in my opinion, and I'm sure you will agree with me, is no longer enough to mitigate losses. Dr. Shahu in the morning mentioned about emergence of so many viruses continuously in fish. What we need is a platform system which you can use it almost like a plug and play. I believe this vector can be used to produce any number of vaccines so long the virus, mRNA in this case or VNN, has cell or tissue tropism for the host that you are targeting. Because using synthetic biology, producing producing a new vaccine would really take no more than 2 to 3 months' time. So, and one last thing I would want to highlight that insect cell culture is well established. Human vaccines are produced with large number of, both animal recombinant protein for animal use, human applications are all there. The downstream pathway to produce large quantity is already established. All we need is to have the right antigen in our hand that can elicit the immune response. And I think that would open up a whole new horizon. I'm thankful to many different agencies that provides funding for our lab, but most importantly, the people who work in our lab. And thank you all. Happy to take any question.
Thank you. Thank you. Very nice presentation.
Thank you.
Probably holds future.
Thank you.
Okay. One question, please.
So thank you very much. Very quick question. You have inactivated the replication machinery, but what about the other genes which cause the infection? If they are all still there, then the presence of any natural infection in a system can provide the missing function, and whatever you have introduced in the shrimp, which is that is the problem with all live attenuated vaccines actually, that the missing parts can be supplied by a natural infection present in the environment. I think Dr. Meka also wants to ask. You also want to ask.
So since we have completely eliminated, you can totally eliminate the RDRP, so there is no replication machinery to support the viral replication. Now, you can say that it can support the replication of RNA2. Sure, it can. But the balance for a virus to make mature virion in a cell, there's a defined balance of the different proteins, means the viral encoded protein. When this balance is disturbed, then you may not get enough mature virion. And if I may add one other thing, you replace the RDRP with targeting RNAi molecules targeting different genes. When you do that, then you can knock out whatever the gene you want to target actually.
Thank you, Dr. Dhar. It was a very impressive talk and a lot of information to get, and I can realize how massive this work would be. I guess it will be around 4 or 5 years hard work will be there in this. Let me understand, we have like baculovirus where full mRNA is created again, and that mRNA is used as vaccine in an oral system, right sir?
Correct.
So here the baculovirus is being used in some kind of source. So what was the source? The cell line, because some cell line would be used for multiplication of mRNA, right? Because ultimately you are using mRNA as the vaccine.
Yes, so basically what we are doing, mRNA on its own cannot infect SF9 cell.
Yeah.
And SF9 cell have been reported to be susceptible to some viruses, but they are not a very robust replication machinery. So what we are doing, because mRNA cannot directly infect in a robust manner, so what we're using is using baculovirus as a way to deliver the payload. And what is the payload? Payload is the RNA1 and RNA2 of mRNA. once that goes there, then the translational machinery supports the replication. That's why when you put the GFP, it clearly shows that it must support the replication of the expression of the foreign gene. So as long as you can overcome the host barrier, so baculovirus is basically enabling us to overcome the host barrier. That's all we are doing.
Yep.
So it's opening up, in other words, producing virus in a non-host cell. Maybe I need to write an article on this.
Yeah.
It's a little bit convoluted way of coming into, but this is going to be a very robust system.
So if I understand correctly, it is like one plasmid, one virus. The dose will be like that. So one plasmid is producing one virus and it is not replicating. So the dose of plasmid will be deciding factor on the dose of vaccine. Will it be like that?
That's correct, yeah.
Okay,
That's
thank you. Thank you so much for making me understand.
Yeah. Thank you.
Thank you, Dr. Dar.
Madam.
Okay, yes, you have opened up a new study completely. Having said that, see, now we are talking about the dominance of non-specific immune response, say in a system like shrimp, okay? But you have brought in the specificity of the immune response.
Yes.
Okay?
Yes, yes.
So now, are you going to tell us that for every virus that affects them, there is going to be the possibility of having a specific vaccine for each of them?
So as long as you know the antigen,
Yeah.
so there's two things needed here in order for a vaccine. So VNN has a wide tissue tropism. It infects a wide number of viruses.
That's for fish, yes.
Yeah, for fish, yeah.
Yeah.
So as long as you have the right antigen and you use the VNN machinery to package it, you can really make any number of vaccine. And not only that, let me just quickly add one other thing. You can modify the RNA2 to make a chimeric particle. You can really make multivalent vaccines in many different combinations, actually.
Yeah, so you chose mRNV1 and the satellite virus.
Yes.
So these are shrimp viruses, the prawn virus, okay?
Correct.
Because white muscle
Correct.
control of that you have shown.
Correct.
But you're talking about the VNN, which is all the viral diseases of the fish.
Mm-hmm.
That is understandable because it's a mechanism that you have pointed out. But if you're thinking about other viruses of, say, shrimps, because that is dominating here for us, if the antigenic component is known, for instance, in white spot syndrome virus, the VP28 is very well studied, along with some others as well, okay? So, according to your studies now, if you are able to selectively include them in the plasmid and deliver to the shrimp, the likelihood of a specific vaccine to white spot syndrome virus is possible?
Absolutely. So I would say for shrimp, because they don't have adaptive immunity, it would be better to use for RNAi. We are actually using it for an RNAi work already.
Yeah, many RNA viruses are there, but it won't be long before you find out something for DNA virus of shrimps as well. So by the time the next DAA is there, you will come out with something on that. Anyway, thank you very much. I think this holds a lot of promise. That's
it holds a lot.
Thank you.
Thank you so much. Thank you.
Thank you. Thank you.
Thank you,
Doctor.